this video, we're going to cover DNA structure and function. We're going to break down how the structure and chemical properties of DNA carry genetic information, how genes are arranged in the long DNA molecule that form each chromosome, and how eukaryotic cells fold and package these molecules into compact chromosomes inside the nucleus. Lastly, we'll take a look at how this packaging is altered to allow access to the underlying DNA for processes like DNA replication and repair. Let's get started. We now know that DNA carries the genetic information of the cell. It's incredible how DNA is a long polymer composed of only 4 types of nucleotide subunit. Yet it carries the genetic material that is copied, replicated, and encodes the instructions for making proteins. Because of its simplicity, biologists in the 1940s actually had difficulty accepting how DNA could be the genetic material. Then in the 1950s, Maurice Wilkins and Rosalind Franklin examined DNA using X-ray diffraction analysis, which is a technique for determining the 3-dimensional atomic structure of a molecule. So DNA was targeted by an X-ray beam, which was diffracted when it contacted an atom, and the pattern was recorded on a film. It was discovered that DNA was composed of 2 strands of the polymer forming a double helix, and it twists at regular intervals. And their results actually provided one of the major pieces of evidence that led to the Watson and Crick's model of the double helical structure of DNA in 1953. And that's how DNA was discovered, the structure of DNA. So let's now subtract complexity and break down the structure of DNA and how it's able to store heredity information. A molecule of DNA, or deoxyribonucleic acid, consists of 2 long polynucleotide chains. Quick revision, a polymer is a long molecule consisting of many similar subunits that are linked together by covalent bonds. Recall what covalent bonds are: a strong chemical bond in which one or more pairs of valence electrons are shared. These repeating units are the building blocks of a polymer, and they are called monomers or subunits. If you've seen the biological molecules lecture, recall that DNA belongs to the class of biological molecules called nucleic acids, and polymers of nucleic acids are called polynucleotides. Now, each chain or strand is composed of 4 types of nucleotide subunits, and the 2 strands are held together by hydrogen bonds between the base portions of the nucleotides. Let's go through the structure of nucleotides. A nucleotide is composed of a nitrogen-containing base or nitrogenous base, a 5-carbon sugar. In DNA, the sugar is deoxyribose, to which a phosphate group is attached. The region of a nucleotide that doesn't have a phosphate group is called a nucleoside. Let's take a closer look at the structure of a nucleotide, beginning with the nitrogenous bases. They can be either adenine, thymine, guanine, or cytosine. Each nitrogen-containing base has 1 or 2 rings that include nitrogen atoms. The reason why they are called nitrogenous bases is because the nitrogen atoms tend to take up protons from solution, and so it acts as a base. There are 2 families of nitrogenous bases, pyrimidines and purines. It's like Romeo and Juliet. A pyrimidine has 1 6-membered ring of carbon and nitrogen atoms. The members of the pyrimidine family are cytosine, thymine, and uracil. Uracil is only found in RNA. Purines, on the other hand, are larger, with a 6-membered ring fused to a 5-membered ring. The purines are adenine and guanine. Okay, so that's the nitrogenous base. The next component is the sugar. In DNA, the sugar is deoxyribose. This sugar lacks an oxygen atom on the 2nd carbon in the ring, hence the name deoxyribose. I've also included the prime symbol to the carbon numbers in the sugar. Okay, so carbon 1, 2, 3, 4, and 5. All right, we now have the nucleoside of a nucleotide sorted. So the next component is the phosphate group, which is attached to the 5' carbon of the sugar. Okay, so we have our nucleotide now. How are then nucleotides linked into polynucleotides? Let's go through this. This involves a condensation reaction. if you recall how polymers are synthesized, it involves two molecules covalently bonded to each other. So quick revision, each monomer contributes part of the water molecule that is released during the reaction. So between these two monomers that are combining, one monomer provides the hydroxyl group, okay, the OH, while the other provides a hydrogen. So this reaction is repeated as new monomers are added to the chain and we make a polymer. And to break down polymers into monomers, this is done by hydrolysis, the reverse reaction or the reverse process of the dehydration reaction. So we're going to be hydrating the polymer and break the bond between monomers. Now, nucleotides are joined by a phosphodiester linkage. The nucleotides are covalently linked together through the sugars and phosphates, which would then form a repeating pattern of sugar phosphate units called the sugar phosphate backbone. Okay? This backbone is called the sugar phosphate backbone, and the nitrogenous bases are not part of this backbone. Now, I mentioned before that the nucleotides are held together, joined by phosphodiester bonds that link the 5' end of one sugar with the 3' end of the next. They are covalently linked through the sugars and phosphates. So the two free ends of the polymer are distinctly different from each other. One end has a phosphate group attached to a 5' carbon, and the other end has a hydroxyl group on a 3' carbon. We can refer to these as the 5' end and the 3' end. The nucleotide runs from 5' to 3' with the bases attached all along the sugar phosphate backbone. Alright, that's one strand. A DNA molecule has two polynucleotide chains or strands that wind around an imaginary axis, forming a double helix. These strands are held together by hydrogen bonding between the bases on the different strands. The two sugar phosphate backbones run in the opposite 5' to 3' direction from each other. This arrangement is referred to as anti-parallel, okay? They run anti-parallel to each other. Now, the sugar phosphate backbones are on the outside of the double helix, and all the nitrogenous bases are on the inside. The bases do not pair at random, okay? The two strands of the double helix are complementary, which means only certain bases are compatible with each other. Let's go through base pairing. Adenine in one strand always pairs with thymine, and guanine always pairs with cytosine. A purine, which is a two-ring base, is paired with a pyrimidine, which is a single-ring base, and each purine-pyrimidine pair is called a base pair, okay? Wow, this is just breathtaking because this complementary base pairing allows the base pairs to be packed in the energetically most favorable arrangement along the interior of the helix. DNA's chemistry is so simple, yet absolutely incredible in the way it's arranged, because this complementarity is important when it comes to copying and maintaining DNA structure. All right, that's the structure of DNA, but how can the structure of DNA, that's so simple, carry the information for specifying an organism, and how can the information be carried accurately? This is amazing. Let's go through this. The information is encoded in the sequence or order of the nucleotides along each DNA strand. We can consider each base, adenine, cytosine, guanine, or thymine, a letter in a four-letter alphabet that is used to spell out biological messages. So just like different languages such as Morse code, English, or a musical notation, in this case, we're just examining DNA, but it's also a language. Organisms differ from one another because their DNA molecules have different nucleotide sequences, carrying out different biological messages. Just like when it comes to songs, it has different musical notations. Now, how can the information be carried accurately though? This is where the double-stranded structure of DNA comes in, because like we've covered, each strand of DNA contains a sequence of nucleotides that is exactly complementary to the nucleotide sequence of its partner strand. So then each strand can act as a template for the synthesis of a new complementary strand. For example, if we have a parental DNA double helix here, both of these strands can serve as a template for its own duplication, producing daughter DNA double helices. The ability of these DNA strands to act as a template allows a cell to replicate its genome before passing it to the next generation. Gorgeous process. And we're going to cover this in detail in the DNA replication lecture. I'm pumped for that one. Okay, going back to the nucleotide alphabet, how is this nucleotide alphabet, which is only 4 letters, adenine, thymine, cytosine, and guanine, used to create biological messages? And what do they spell out exactly? Let's go through this. A basic unit of inheritance is known as a gene, and this is what determines the amino acid sequence of a polypeptide, and genes consist of DNA. Essentially, it's a segment of DNA that contains instructions for making a particular protein. Why am I bringing proteins into this? If you've seen the previous lecture on proteins, we covered how proteins are the cell's building blocks and how they execute majority of the cell's functions, and how there are different types of proteins that have different specific functions, such as enzymes, structural, storage, motor, and specialized proteins that can act as antibodies or hormones. So DNA information is able to encode proteins. The complete store of information in an organism's DNA is called its genome. Now, the function of a protein is determined by its 3-dimensional structure, which in turn is determined by the sequence of amino acids in its polypeptide chain. If you'd like a refresher on this, watch the previous lecture on protein structure, function, and regulation. So the linear sequences of nucleotides in a gene spells out the linear sequence or order of amino acids in a protein. Because the sequence of amino acids is not random, it's inherited genetic information, and nucleic acids store, transmit, and help express genetic information. Absolutely beautiful. It's magic, okay? This is where the genetic code comes in, which is the set of rules by which the information contained in the nucleotide sequence, the 4-letter alphabet A, T, C, and G, of a gene is translated into the amino acid sequence of a protein, the 20-letter amino acid alphabets of proteins. We're going to cover the genetic code in detail in the transcription and translation lecture, or gene expression, when we break down how the nucleotide sequence of a gene is transcribed into the nucleotide sequence of an RNA molecule and then translated into the amino acid sequence of a protein. So we'll cover how we go from DNA to RNA and then from RNA to proteins. All right, we've covered a lot so far, but before we move on to how large amounts of DNA is packed neatly in the cell nucleus, let's do a quick review. So genetic or heredity information is carried in the linear sequence of nucleotides in DNA. A nucleotide is composed of a nitrogen-containing base or nitrogenous base, a 5-carbon sugar to which a phosphate group is attached. Okay, a molecule of DNA consists of 2 polynucleotide chains or strands, and these are complementary anti-parallel chains of nucleotides held together by hydrogen bonds between the nitrogenous bases. Adenine in one strand pairs with thymine in the other, and cytosine pairs with guanine. Okay, in eukaryotes, DNA is contained in the cell's nucleus, All right. Let's now break down how DNA molecules are packaged neatly into chromosomes. An enormous amounts of DNA are required to encode the information to create a unique multicellular organism. And the most crucial function of DNA is to carry genes. Genes, like we mentioned before, are the basic and functional units of heredity. A gene is a segment of DNA that contains the instructions for making a particular protein or RNA molecule. And this information is located in the cell nucleus. In eukaryotic cells, DNA molecules are packaged into chromosomes. These chromosomes are then duplicated and apportioned between the 2 daughter cells at each cell division. We're only going to be talking about eukaryotic organisms, but quick review, in prokaryotes, bacteria carry their genes on a single circular DNA, and they also lack a nuclear compartment. So let's go through how DNA is packaged into multiple chromosomes. I say multiple because DNA is distributed among a set of different chromosomes. Each chromosome consists of a single long linear DNA molecule associated with proteins that fold and pack the DNA into a more compact structure. Chromosomes are also associated with many other proteins and RNA molecules that are required for gene expression, DNA replication, and DNA repair. Okay? The structure of DNA and tightly bound protein is called chromatin, and we'll go through this in a second. Referring to human cells, each human cell nucleus contains 2 copies of each chromosome, one inherited from the mother and one from the father. This doesn't apply to gametes though, sperm and eggs, and also highly specialized cells that lack DNA, such as mature red blood cells. The maternal and paternal versions of each chromosome are called homologous chromosomes or homologs. The only non-homologous chromosome pairs in humans are the sex chromosomes in males, where a Y chromosome is inherited from the father and an X chromosome from the mother. Whereas in females, they inherit 1 chromosome from each parent, so there are 2 X's. And they don't have a Y chromosome. So then each human cell contains a total of 46 chromosomes, 22 pairs that are common in both males and females, called autosomes, plus the 2 sex chromosomes, X and Y in males and 2 X's in females. So then how can we tell or distinguish these sets of chromosomes from one another? One thing is that they are all different sizes. We can also paint each chromosome a different color using a technique called special karyotyping based on DNA hybridization, where a short strand of nucleic acid is tagged with a fluorescent dye that serves as a probe that picks out its complementary DNA sequence, lighting up the target chromosome at any site where it binds. This is usually done during mitosis when chromosomes are compacted and easy to see. Another method is to stain them with dyes that reveal a distinct and reproducible pattern of bands along each mitotic chromosome. And the patterns of band on each type of chromosome is different from one another. So we can use that to identify and number each chromosome, okay? Human chromosome. This here show the banding patterns of human chromosomes. An ordered display of the full set of 46 chromosomes is called the human karyotype. If parts of a chromosome are lost or switched between chromosomes, these changes can be detected. Absolutely incredible. And we can use karyotypes to detect chromosomal abnormalities that are associated with some inherited disorders. Okay, so chromosomes organize and carry genetic information. Specifically, chromosomes carry genes, the basic functional units of heredity. And recall that the total genetic information present in an organism constitutes its genome. Now, only a small amount of the human genome, few percent, codes for proteins. The coding sequences are called exons, and the non-coding sequences in genes are called introns. We'll talk about this in another lecture, okay? Now, each gene is associated with regulatory DNA sequences, which are responsible for making sure that the gene is turned on or off at appropriate time and expressed at the right level in the appropriate type of cell. All right, now that we know that chromosomes carry gene and we have 23 pairs of chromosomes, 22 pairs are called autosomes plus the sex chromosomes, how do you actually form a functional chromosome? Because a DNA molecule simply just can't carry genes. It must be able to replicate, and the replicated copies must be separated and partitioned equally into 2 daughter cells at each cell division. This process is known as the cell cycle, and we'll cover this in detail in another lecture. In this lecture, we're just going to focus on the interphase here, when chromosomes are duplicated, and also the M phase or mitosis, when the chromosomes are segregated to the 2 daughter nuclei. During interphase, this is a long process, genes are expressed and DNA replication occurs. Now, I mentioned before that a DNA molecule, more specifically chromosomes, must be able to replicate, and the newly replicated copies must then be separated and partitioned equally into the 2 daughter cells. These processes or functions are controlled by 3 types of specialized nucleotide sequences in the DNA that ensure that this entire process occurs efficiently. Let's draw this out. Our chromosomes here, okay, are at different stages of the cycle. One type of sequence is called a replication origin. This is where DNA replication begins. The cool thing about eukaryotic chromosomes is that they contain many replication origins. So we have one here and then another one there, and this is to allow DNA replication to occur rapidly. After DNA replication takes place, the two sister chromatids that form each chromosome, they're still attached to one another. And as the cell cycle continues, they become more compact or condensed. And the highly condensed chromosomes are known as mitotic chromosomes. Once they become more condensed, one copy of each chromosome will be pulled into each daughter cell when a cell divides. And this is where the second nucleotide sequence comes in, called a centromere. The centromere helps to hold the duplicated chromosomes together and allows the mitotic spindle to attach to each duplicated chromosome, and they are separated to each of the two daughter cells. How are the duplicated chromosomes attached to the centromere? A protein complex called a kinetochore forms at the centromere, okay? And it attaches the duplicated chromosomes to the mitotic spindle, allowing them to be pulled apart. Now, the third specialized DNA sequence forms the ends of a chromosome, called telomeres. Telomeres also contain repeated sequences that are needed for the ends of chromosomes to be fully replicated. They also protect the end of the chromosome from being mistaken by the cell for broken DNA in need of repair. And we cover this in the DNA replication and repair lecture. It's absolutely beautiful how all these lectures connect together. Okay, so these are the three specialized DNA sequences required to produce a functional chromosome that can be replicated and then segregated. We have the replication origin, the centromere, and the telomeres. Now, comparing interphase chromosomes to mitotic chromosomes, they're much longer and finer. But even though interphase chromosomes are less dense than mitotic chromosomes, they are still tightly packed and more extended. And mitotic chromosomes are highly condensed, and this is caused by proteins that coil and fold the DNA into higher levels of organization. So let's break down how these specialized proteins make this possible. All right. The proteins that bind to DNA to form chromosomes are divided into two classes, the histones and the non-histone chromosomal proteins. Both classes contribute about the same mass to a chromosome as DNA, and along with nuclear DNA, this is known as chromatin, okay? Both classes of protein with nuclear DNA is called chromatin. Let's take a closer look. Histones are responsible for the first and most basic level of chromosome packing, the formation of the nucleosome. Nucleosomes convert the DNA molecules in an interphase nucleus into a chromatin fiber. So if we were to break open interphase nuclei and examine these chromatin fibers under an electron microscope, we'd find these fibers contain clusters of closely packed nucleosomes. Now, if chromatin is experimentally unpacked or unfolded slightly, it can be seen in the electron microscope as a series of beads on a string, or the way I like to look at it, macarons on a string, specifically salted caramel macarons on a string. The string is DNA, and each bead is a nucleosome core particle, okay? So the string here is DNA, and each bead, or each macaron, salted macaron, is a nucleosome core particle that consists of DNA wound around a histone core. Looking at chromatin here, which consists of, again, DNA bound to both histone and non-histone proteins, how do you determine the structure of the nucleosome core particle? Let's break this down even further. Let's go through this. First, researchers treated chromatin in its unfolded macarons on a string form with enzymes called nucleases that break down DNA by cutting between the nucleosomes, okay? Breaking the phosphodiester bonds between nucleotides. After digestion, the exposed DNA between the nucleosome core particles, called the linker DNA, is degraded, and the core particles are isolated. So we have an isolated macaron here. Now, an individual nucleosome core particle consists of a complex of 8 histone proteins, 2 molecules each of histones H2A, H2B, H3, and H4, as well as the double-stranded DNA that is 147 nucleotide pairs long. The histone octamer forms a protein core around which the double-stranded DNA is wound, okay? The linker DNA between each nucleosome core particle can vary in length, from a few nucleotide pairs up to about 80. So looking at the structure of the nucleosome core particle here, we can see how DNA is packaged. The nucleosomes are further packed on top of one another, instead of just the macarons on the string. And this is to develop a more compact structure, okay? So let's go through what causes nucleosomes to stack so tightly on each other. This additional packing of nucleosomes into a chromatin fiber depends on a fifth histone called histone H1. This linker histone binds to each nucleosome and it's going to change the path of the DNA as it exits from the nucleosome core, allowing it to form a more condensed chromatin fiber. Essentially, histone H1 pulls nucleosomes together into a regular repeating array, okay? Now, to produce an even more condensed structure, a specialized non-histone chromosomal proteins fold the chromatin into a series of loops. And we call that mitotic chromosomes are more condensed than interphase chromosomes, so the string of loops undergo at least one more level of packing. Okay, we've covered a lot so far, so before we move on, let's do a quick recap on how DNA packing occurs on several levels in chromosomes. We have a DNA double helix here, and the two types of protein involved in chromosome packing, along with nuclear DNA, is called chromatin, okay? And histones are responsible for the first and basic level of chromatin packing, which is the formation of nucleosomes. Nucleosomes convert the DNA molecules into a chromatin fiber. And if we were to partially unwind or unfold chromatin, it can be seen as a series of beads on a string, but we're calling them macarons on a string because it's more fun. Specifically, salted caramel macarons. So the string is DNA, and each macaron is a nucleosome core particle, which consists of DNA wound around a core of histone proteins, okay? Then the nucleosomes are further packed on top of one another and folded into loops to produce a more compact and condensed structure. And then we have our entire mitotic chromosome. Recall that histone H1 and a set of specialized non-histone chromosomal proteins help create these loops. Alright, so we've covered how DNA is packaged into nucleosomes to create a chromatin fiber. Let's now go through how different chromatin structures are created in different regions of a cell's genome. Recall that mitotic chromosomes are more condensed than interphase chromosomes, but interphase chromosomes contain more extended forms of chromatin. Okay, now there are regions of the chromosome which contain genes that are being actively expressed and they are generally more extended. And regions that contain silent genes are more condensed. So the structure of an interphase chromosome can differ from one cell type to the next, helping to determine which genes are switched on and which genes are switched off. All right, let's break this down further. There are two types of chromatin in the interphase nuclei, a highly condensed form called heterochromatin and all the rest, which is less condensed, called euchromatin. Heterochromatin typically makes up about 10% of an interphase chromosome and is concentrated around the centromere region and in the telomeric DNA at the chromosome ends. Okay? Oh, and I said that condensed areas contain silent genes, but these aren't dead DNA, okay? These areas are generally just unusually resistant to gene expression. So they're not dead, they're just silent. Okay, so those are the two types of chromatin. Each type are established and maintained by different sets of histone tail modifications, which attract a distinct sets of non-histone chromosomal proteins, okay? For example, let's slow down here. Let's take a look at the formation of the most common type of heterochromatin. It includes the methylation of lysine 9 in the tail of histone H3. And once heterochromatin has been formed, it can spread to neighboring regions of DNA. Let's break this down further. Okay, we have our heterochromatin and euchromatin here, the heterochromatin are more condensed. Histone tail modifications attract a set of heterochromatin-specific proteins, including histone-modifying enzymes. That's a mouthful. Okay, so these blue things here are heterochromatin-specific histone tail modifications, and they are going to attract a set of heterochromatin-specific proteins, including histone-modifying enzymes. So let's bring these bad boys in. So here are the specific proteins that are going to reproduce the same histone modifications on neighboring nucleosomes, okay? Nearby histones. And these modifications will continue, and the extended region of heterochromatin will continue to spread until it encounters a barrier DNA sequence that stops the distribution, okay? So this is a barrier DNA sequence, this purple line here, and it's going to block further recruitment. So heterochromatin spreads until it encounters a barrier DNA sequence. Now, majority of DNA that is folded into heterochromatin does not contain genes, okay? And when genes are accidentally packed into heterochromatin, it usually fails to be expressed because they are silent genes. Okay, we've covered how DNA is packed into chromatin. Let's now go through how this packaging can be adjusted to allow fast access to the underlying DNA. Let's break down how a cell can alter its chromatin structure to expose localized regions of DNA and allow access to specific proteins and protein complexes for processes like DNA replication and repair. There are several ways of altering chromatin structure, which involves chromatin remodeling complexes and histone modifying enzymes. But in this lecture, let's just go through ATP-dependent chromatin remodeling complexes. These protein machines use the energy of ATP hydrolysis to change the position of the DNA wrapped around nucleosomes, okay? So how it works is these complexes interact with both the histone octamer and the DNA, and it's going to change the arrangement of the nucleosomes. It locally repositions the DNA that's wrapped around nucleosomes so that the DNA is more or less accessible to other proteins in the cell. And during mitosis, many of these complexes are not active, which may help mitotic chromosomes maintain their packed structure. Okay? Let's go through this again. So we have our salted caramel macaron here, or nucleosome. The string is the DNA that's wrapped around the histone octamer, and this here is the ATP-dependent chromatin remodeling complex. The complex uses energy that's derived from ATP hydrolysis to loosen the nucleosomal DNA, and it's going to reposition it. These stripes here that I've drawn is to show you how the DNA shifts its position. And many cycles of ATP hydrolysis are required to produce such a shift. So then the DNA is more accessible or less accessible to other proteins in the cell, okay? Now, we won't go through histone modifying enzymes, but both histone modifying enzymes and chromatin remodeling complexes work together to condense and relax regions of chromatin, allowing local chromatin structure to alter rapidly according to the needs of the cell. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video, or you can view the entire playlist. Hey, stop procrastinating!